Hello and very good afternoon to all of you who have been joining us in this webinar. My name is Irene and I'm the application specialist and certified trainer for Apical Scientific. So today in this particular webinar, I will be talking on the technologies and challenges that is involved in metagenome sequencing. So this is the scope of my talk, as you see. So in this particular talk, I'm going to introduce to NGS, especially focusing on metagenomic. Okay, so under the umbrella of metagenomic, I will talk on Amplicon sequencing as well as shotgun sequencing, and also the challenges that is involved involving these two NGS platform. And finally, I will close with conclusion. Now, first, let's take a look into what is NGS. So NGS is actually an abbreviation to next generation sequencing. So as per its name, this technology came in as a second generation after Sanger sequencing. So this technology is actually able to perform a million sequencing reaction simultaneously. Okay, so because it's able to do that, so it is also referred as a massively parallel sequencing. Okay, so this is the two things that NGS has, the specialty of it. So NGS also consists of several technologies that uses distinctive chemistry. Now, for example, we have Illumina, we have PacBio, we have Ion Torrent, we have Nanopore. So for NGS, one can actually use either DNA or RNA to sequence. So if you're actually using RNA as a sample, so what you can do is basically you can convert it to cDNA, and the library preparation which is made for both DNA and RNA is in the base of DNA. So the cDNA is actually converted to DNA, and then so the cDNA and this library is being prepared. Okay, so the main platform that we have, okay, there are two main platform that is very much common, which is the Illumina and PacBio. So besides Ion Torrent and Nanopore, we also have Illumina and PacBio. So as for these particular two platform above, which is the common one, which is the Illumina and PacBio, the sequencing approach that is used is actually sequencing by synthesis. And the purpose they do this is actually to generate multiple clusters. So NGS also has its own advantage, especially with its ability to give a high throughput sequencing read. So not only that, it is also easily scalable with a shorter runtime. Now, this is the types of NGS that we have. So what I'm going to do is I'm actually going to focus on metagenomic. But before I move on to metagenomics, let me give you brief description or brief introduction on the type of NGS. Okay, so the first thing is the circle in brown, so the whole genome sequencing. So this whole genome sequencing is used to sequence the whole genomic region of an organism. So it can be a DNA or an RNA. So any genome, okay, DNA base of an organism that you want to sequence. So your approach should be for whole genome sequencing. And if you're going to do whole exome sequencing, so this is actually a technique to sequence all protein coding regions of genes in a genome. Okay, so then you tend to focus on exome sequencing. For single cell, as per its name, it is to sequence individual cells to understand better on the function of a cell. So this is why single cell sequencing comes into play. Then we have ChIP-seq. So ChIP-seq is actually a method to analyze protein interaction with DNA. So you want to identify DNA binding sites. So this is where ChIP-seq comes into the play. Bisulfite sequencing is to determine the pattern of methylation. Now, for this particular study, it's very common to do in a human. All right. And then we have targeted gene sequencing, which is actually very much specific to targeted area of a microbes. Okay, so for example, if you want to do ITS or you want to do 16S, it's very much targeted site. And for RNA-seq, what we have here is you can sequence the total RNA of an organism at a given moment. So this is the types of NGS that is available. Now let's move on to metagenomics. Now in this particular webinar, we will focus on metagenomic. So what is actually metagenomics? So this metagenomics is actually a broad field which can be used to study environmental genomics, ecogenomics, or even community genomics. So there are actually two types of platform that you can use to study metagenomics. One is actually the Amplicon sequencing, and another is actually using shotgun sequencing. So under Amplicon sequencing, the most common technology used is Illumina. So when it comes to shotgun, the most common one is either Illumina or PacBios. These two are the most common one. So let's look into detail for Amplicon sequencing. So what is Amplicon sequencing? So why is it called Amplicon in the very first place? So Amplicons are actually PCR amplified DNA products. So basically they are actually short fragment of a specific genomic region. So, and this particular region is actually obtained by PCR, the specific region. So this specific region can be the identification region. So the identification region is where 16S comes in, ITS comes in, and 18S. Okay, so now Amplicon sequencing can be used for any mixed samples, any mixed species sample, and with that particular mixed samples, you actually can target one specific microbe group. So for example, if you have a soil, so soil consists of multiple microorganisms, like you have bacteria, fungus, right? Now, if your main aim is to target for bacteria, then you use a very specific identification region, which is the 16S, to target for bacteria. So what we are seeing here is basically at the bottom here is the microorganism, like what I've explained earlier. You have bacteria, fungus, archaea, as well as protozoa. So these are the microorganism, and some of the primers are basically the identification region. They are not primers, they are identification region to identify these particular species. Okay, so how do we do it? Okay, so the first thing is to isolate genomic or DNA template from the sample. So these samples can come from environment-based, ecology-based, or community. For example, you want to study on human gut, animals, tools, and etc. Then what you can do is you have to use a gene-specific primers to amplify and create amplicon. So upon amplification, so these amplicons are basically indexed. So you can see in the picture that, you know, there's what we call these primers, and this at the end here, it's where usually these particular fragments will be indexed. Okay, and then samples will be pooled, and then we prepare a library by adding adapters and enriching the target regions. So why the reason why we add adapter is basically we want to make sure that this particular amplicon that is being indexed, they tend to adhere to the flow cell for sequencing. So once sequencing is done, so all the raw reads, they'll be trimmed. We align them to obtain full sequence that was amplified earlier. Okay, so it has to be similar. So as I explained earlier, the regions that are used to identify the microorganism is as listed on my right. So these are the region. Okay, so if you see this particular primers, these are the primers that basically our NGS service, you know, we tend to offer. So there are few regions here. So if you have any other regions rather than what is stated here, you can always get in touch with us. Okay, you can always get in touch with us. So we have the 16S, 18S, and ITS region. So anything rather than this, you can always get contact with us. So having this, now the next question you may ask is, how do I know which region should I use or should I choose? So the next thing is, the most important part is to understand the objective of your study. So where and what are you sampling, as well as what is the target species that you're looking for? Is it bacteria? Is it fungus? Or is it algae? So find a reference paper that is close to the nature of your study. For example, what you're studying now could be a swamp forest, okay, and the objective you want to look into is the bacteria distribution. So find for a close paper. So in this particular slide I have attached, okay, a Malaysian tropical peat swamp forest. So you can use this as a reference. And then you want to know what kind of region did they use to identify the bacterium. So scroll to materials and method. Look for what region do they use. So if you see here, I have underlined in red V4 region for 16S. So you can use this particular region to sequence. Now, before even you choose this region, you also need to check, okay, when they use this region, what species is found abundance or rich in that particular area. Okay, so if these are some of the microbes of your target, then you can use this particular region to sequence. Now, in case you are not sure, you do not know what kind of species I may even get, then you can use this particular region, and once you have all your sequencing data, your bioinformatic analysis, so you can do a discussion. For example, maybe this peat swamp that you want to analyze is in Thailand, so you can do a comparison studies. Maybe in Malaysia it's like this, so the distribution of bacteria in Thailand could be different. So you can use this as a reference paper and you can compare. So similarly, I've also attached two more papers here. That is one for rice, and another one that is actually looking into community ecology. It covers bacteria, archaea, microbial. So they are also having the region. So it's good to look into these regions. Yeah. Now, another question that is very common researchers tend to ask is, how do I know how much of raw data or number of reads should I choose? Okay, so why this is actually very important? Because the number of raw data that you are going to choose also determines the cost that you're going to pay for this particular NGS service. So first and foremost, find a reference paper that is close to the nature of your study, or maybe it has a similar experimental design. Okay, so what are you ought to check is to check for the rare fraction curve. So usually the rare fraction curve will be in the result site. So look at the rare fraction curve, which they have attached in this particular paper. So if the curve has not reached plateau, okay, plateau means is basically the the range where they do not climb anymore, they are much more static. Okay, so if they do not have insufficient data, what happens is the rare fraction curve, the curves will be still climbing. Okay, so which means you need more data for it. So there's actually insufficient data and more data needs to be sequenced. So how much of data do you just have to add? So you can add approximately 100K reads. That is more than enough. Okay, and then you can sequence, and then upon bioinformatic analysis, you get to see whether upon sequencing, the latest sequencing that you have done, is it nearing plateau or not. So if it's actually nearing, okay, if that particular journal reports, okay, it's using this particular region, this is what rare fraction curve that you're getting. So you have enough confidence that your data is very much enough. So which means because it's already aiming to plateau, so you do not need to add any more data. So on my right is what you see, the NGS services that first base offer. And also I've highlighted the data output. So we can go up to 200 raw reads, and we also give an option to you whereby you can do 200 raw reads, 100K raw reads, or 60K raw reads. So choice is on you, and you make the choice based on the rare fraction curve. Sample type and preparation. So how do you prepare samples. So first and foremost is to isolate DNA, because both these NGS actually utilizes DNA from the samples. So you run gel electrophoresis, make sure you have an intact band or basically bright DNA band, quantify the concentration, and then you send for sequencing. Also, please be reminded that you need to also identify the region that you want to sequence. So based on the information that I shared earlier, so you can actually determine which region do you need, which target microbes are you looking for. So this information need to be specified before you send your samples for sequencing. So on my right, what you're seeing is gel images, okay, gel agarose images of DNA that has been extracted, quantified, and now we run on gel. So basically this, the first image is, we encourage, which is accepted for NGS sequencing. So this is accepted. But on the other hand, the three particular gel agarose, we do not encourage for NGS sequencing, but we also understand that some of these particular samples, okay, may be precious to you, especially samples that you obtain from patients, natural environments like corals that you cannot go and sample often. So in this case, what we do is we do not reject these samples, but we just want to let you know that these particular samples possesses risk. Okay, so now for this particular Amplicon sequencing, because the library preparation is made by amplifying through PCR, so the success rate usually, so it's very depending. So on the Amplicon library preparation, so when you give us, DNA's DNA template like this, so the success rate is usually from 60 to 80%. So it is very much depending on the Amplicon DNA library preparation. All right. So we do not reject, but it's just that perhaps the risk is something you have to acknowledge on that part. Yeah. Of course, then we go into sample requirement. So this is actually the sample requirement for first base for Amplicon sequencing. So what we actually strongly recommend is approximately 1.6 microgram of DNA in 30 microliter of volume. So it is 1.6 microgram total DNA in 30 microliter. So maybe for each microliter, it's approximately 50 nanogram. So we also recommend that you take OD, and the purity that we encourage should be approximately from 1.8 to 2.0. So at the bottom is what you see, the PAC biotechnology. Okay, the amount of DNA that is needed, and also the volume that is being adjusted accordingly to the amount. Now, let's move on to another part of metagenome sequencing, which is known as shotgun sequencing. So, shotgun metagenomic is an approach used to identify and characterize microbes directly from their natural habitat. So literally it involves all microbes inside. Okay, so the DNA that is extracted from these samples, okay, they are all subjected for sequencing. So this approach is basically very suitable to study population of microbes, for example, in ecology, environmental conservation, infectious disease diagnosis, environmental remediation, biotechnology, and also agriculture. So it can also be used for research as well as diagnostic approach. So it's very much similar to Amplicon sequencing as well, whereby Amplicon you can also use for diagnostic approach. Now, there are few technology and chemistry that can be used to do metagenome sequencing. Okay, but in this talk, I'm just going to focus on the two most common technology, which is the Illumina and PacBio. So Illumina basically produces short reads, while PacBio provides a longer read. So you can choose either one, depending on the objective of your study and also the available budget. Okay, so basically, on my right, you see what's the differences. So the sequencing chemistry for both PacBio and Illumina is the same. They sequence by synthesis, but the difference here is the sample library. So one uses Smart Bell template, while the other, which is the Illumina, it uses double-stranded DNA with adapters. So if you can see data output, Illumina has higher data output. Okay, PacBio is more costly than Illumina. Read length for PacBio is much more longer, that is approximately 14 kb, when Illumina it's approximately 100 to 300 base pair. All right, and PacBio has a higher rate compared to higher error rate compared to Illumina. Now we have seen two types of technology, which is the Illumina and PacBio. So how do I know which technology to use? Okay, so Illumina basically creates short reads. So because it is short reads, so it has a better depth coverage. Okay, but for PacBio, it is longer reads and it has better coverage of the sequence. Now, if you take Illumina, it has a lower error rate. Because why? It has multiple alignment, because it's if you know, it generates clusters, right? So it's able to do multiple sequences, that is sequence, and when you align them, it actually reduces the error rate. Okay, and then for short sequences for Illumina, some of it may not even surpass QC. Why? The reason is because this short read sometimes may be even shorter. So in the first streaming part itself, some of these particular sequences are lost. So in that case, perhaps some of these information of the genes also you may lose. Okay, and Illumina is definitely cheaper in cost. Now, when we look into PacBio, PacBio does not need any assembly because of its larger length that it can sequence. So literally you have information sufficient for many genes. So because there's no assembly, all right, so it tend to give higher error rate that is coming from the sequencing itself. Okay, again, because it's longer read, so most of the genes are included, therefore it gives a good coverage. But PacBio always is much more costly. So on my right, you see here is what First Base offers. Okay, so when we offer for metagenomic sequencing using shotgun method, okay, so what we are giving here is basically we are using Illumina HiSeq or NovaSeq. That's approximately 150 pair N run, and we have two options for data output, which is the 6 gig and 12 gig per sample. So when you want to choose, you definitely need to know the objective of your study. Okay, is there a need for you to know the full sequence of a microbial species? Do you want to study function of it, or will you maybe you want to look into some novel microorganisms? So if these are some of the objectives, then yes, metagenomic is the approach for you. Okay, and again, I would definitely suggest find a reference paper that may help you with these particular reference paper that is very much closer to your study. So again, this is very much similar to what I've explained earlier in Amplicon sequencing. So for metagenomic sequencing samples, usually the DNA that is obtained do have smearing. Therefore, it is actually encouraged to get good quality DNA. But again, like what I've said earlier, we do not reject, because we understand that some samples are precious and you can't sample often, or you cannot go back sampling again. But I would like to let you know that these particular samples possesses risk. Okay, so if you can accept the risk, okay, the risk is ranging from 60% to 80%. So these samples can be sequenced. Now for metagenomic sequencing, the library is directly prepared from DNA. Okay, so there is no PCR. So in this case, like degraded DNA, these kind of samples will be definitely rejected. But if you have samples like, you know, with the presence of very UVC and ban of DNA bands, so perhaps we can still proceed, but it comes with a risk. Okay, so this is the sample requirement for first base for shotgun sequencing, which is very much similar to Amplicon sequencing. So we have seen two types of metagenomic sequencing. So usually those two, they tend to have similar challenges. So let's look into what are the challenges commonly faced by researchers. So number one, how are we going to obtain an intact band? So the first thing is, how do you handle your samples? How do you store your samples? So upon sampling, whichever samples that you are sampling, freeze the samples using liquid nitrogen or store them at -80. Okay, so you freeze and or you can do it as or. So upon freezing the samples, you need to process as soon as possible. Why? Because samples as itself has presence of nucleus. So these particular nucleases can actually degrade DNA and RNA. Okay, because you are freezing them, literally there's nothing that will be alive. So now the nucleases comes active. Okay, they'll start degrading DNA and RNA. Also, you have to give you need to also look into the storage container and bags, because you do not want to introduce RNA or DNA into the particular samples. Next is choosing the right DNA extraction kit. So the example that is shown below is one of the brands that we use in our lab. Okay, so we do use many other, but this is example of what we use. So usually for metagenomics or Amplicon, the common DNA extraction for environmental samples, okay, environmental or community or ecology, it's either soil or stool. Okay, if you want to ask me for my sample, which one should I use? I cannot be buying two, na? So which one should I use, soil or stool? So my suggestion would be to get a sample or trial kit. Okay, so not many distributor or vendor could actually supply samples for you, but you can ask them if they can give you samples, or perhaps you may want to buy a smaller size reaction kits to try out. Okay, so when you try few samples and look at their DNA profile. So you do not want to waste the samples that you have sampled earlier, right? So you just try few and look into their DNA profile. Now, in an situation whereby you already have kits, for example, for bacteria, fungi, very specific DNA extraction kit, I would suggest the same thing. Try out a sample kit, or maybe try out few of your samples, okay, with these particular bacteria, fungal, DNA extraction kit, and check for the DNA profile. So there might be some optimization that is needed as well down the line. So you need to optimize them, okay, and then try to obtain a good and intact DNA. So the third one is to run gel agarose. So this is quite a common mistake where a lot of researchers tend to do, whereby after extracting DNA, what they do is they just do a nanodrop reading. Okay, so and then after nanodrop reading, they just send the samples. So when you send the samples here, we will see a lot of DNA profile that is coming out, especially degraded DNA. Okay. So when it comes to degraded DNA, you will know that this particular DNA, over time, literally, we will not even get anything out of it, because when you want to prepare a library and then we want to sequence them, so basically, especially when it comes to metagenomic, because we're not amplifying, so this particular degraded DNA is not going to give you much information anymore. All right, so it is good to, once you have extracted, run gel to verify. Keep this as a practice because it literally save your time and cost. So another common things that you also will see is DNA with protein contamination and DNA with RNA contamination. So how do you identify them? Protein contamination, usually you will see them in the wells. So when you run DNA, you'll see a protein contamination in the well itself. Okay. Then as for RNA, you will see them at the bottom. So this is the common DNA profile of the gel images. Okay, so RNA will be at the bottom or the middle, while protein usually stuck inside the well. Now, how do I clean up these particular samples? If you have RNA contamination, clean up using an enzyme that is called RNase A. So our experience is not to clean up in spin column. Now, if you remember, most of the kits actually comes with spin column. Okay, and then they will have an optional whereby you can actually clean up your samples in the column itself using RNase A. So our experience thus far is when we clean up inside the spin column, usually we see smearing, DNA smearing. So what we do, usually we will extract and we clean up after the extraction. Okay, now you also need to note that when you're cleaning up, there will be loss in DNA yield. So make sure you have enough DNA concentration and volume in the beginning itself before even you start up with the cleanup. Okay, so once you have done all the cleanup, do check on the quality, means you run the gel again and quantify the DNA. So as for protein, you clean up using phenol chloroform isoamyl alcohol, and ratio that you usually that is been followed is phenol 25, chloroform 24, isoamyl alcohol 1. So basically you can scale up based on this particular ratio. So again, avoid cleaning up in the column. And when you want to do when you want to do a cleanup, there will be also loss in DNA yield. So make sure you have enough DNA concentration and volume in the beginning itself. And again, run the gel to check on the quality and quantify the DNA. Now, technology platform. How do I know what to choose? Do I need to, how do I know whether I want Amplicon or shotgun metagenomics? So know the objective of your project. Okay, are you interested to analyze one type of species, or are you interested to analyze all involved species and the function? So this is the first thing that you need to get things right, and then do enough literature study. So for example, you have an animal gut study. Okay, you want to study animal gut. So you know when it comes to animal gut, there'll be high population of bacteria and lesser in fungi. So unless your objective is to figure out what kind of fungus, you know them very clearly, your objective is, then yes. But if you're if you just want to study what is the bacteria, but you use a different targeted primers, then perhaps you will not get what you want. So that is the reason why you need to do a good literature study, know what is the region that you want to know, to sequence, what is the microbe that you want to look into. So these are very important ones. And then another example like seawater. Okay, so when it comes to seawater, you tend to have population of bacteria, fungi, algae, zooplankton. Metagenomics, basically, you are going to sequence everything that is inside the seawater. But when you're going to do Amplicon, then you are targeting. So maybe you want to study bacteria in seawater, you focus on Amplicon. Now, when you want, we also have targeted based primers. Means, for example, in animal gut study, you want to know a very narrowed species, example Bacillus. So if you have a targeted primer that is going to target on Bacillus, okay, so the best thing before you do for NGS is to confirm the species specificity. So perhaps maybe you want to amplify using PCR, confirm them with Sanger sequencing prior to NGS. All right, so these are some of the tips that could help you. Cost. Cost is also another major challenge. But before even you come to the cost or planning for cost and budget, consider the number of samples and replicates that you need. Now, what is the most important replicates that you need is the biological replicates. Because for with biological replicates, you want to get a meaningful results. Biological replicates helps to see the consistency. Basically, it reduces the variability and gives you a true and valid results. Now, if you want to publish your results, your finding in a reputable journals like Q1, you at least need 3 biological samples per treatment. So recommended is 3. You can go up to 6. So with that, you can plan the cost. So how many samples do you need to send for your sequencing? Will this cover your objective of your study? And now, do you have enough budget? So you can now plan your costing based on the number of samples and replicates. Next is bioinformatics. How do I choose what kind of analysis do I need? So number one is to read a literature review. So find a paper that is close to the nature of your research. Okay. Now, what you need to focus on is on the type of analysis and not based on attractive graphical image. Okay, so what is more important in Amplicon sequencing? You definitely need to know, you need to provide the sequencing QC, raw data, and then you want to look into the alpha beta diversity, species distribution. When it comes to shotgun metagenomic sequencing, you also want to look into the sequencing QC, functional annotation, and also some comparative analysis. So keep it simple for your readers to understand. All right. Now, another key point when you choose bioinformatics is, will this meet your objective? So you need to understand your project thoroughly. What do you want? So pick the right analysis according to the objective of your study. Now, usually for NGS service provider, now, usually they will also provide you with a bioinformatic analysis. So when they provide you with bioinformatic analysis, usually there will be a lot of analysis that has been done. Okay, so and then they'll provide you with the images, the graphs and things like that. So you do not need to use all the information that they have provided. So you just need to pick the right analysis, the right graph to um, to complete or basically to comply with the your study. Okay. On the other hand, you can also customize the analysis according to the objective. So maybe you want to compare just 2 groups. All right. You can also do the customized analysis. So you can always talk to us and we can do this custom based analysis for you. So on my final, what you see is the example. So this is just a very brief example. So on the top box is what you see is for Amplicon. So these are the sum of the graphics. Okay, that is kind of important that you can actually put it into your papers. So for example, you have the species distribution, PCA, PCOA, sample clusters tree, and also Rna display. So these are very common. Okay, very common. So you can use this also for your paper. Now, when you want to do for shotgun metagenomic sequencing, then what you are going to have is basically the pathway comparative study. So this pathway comparative study is basically talking on the genes. Okay, what kind of genes that is involved, because it is actually on the functional analysis. So you get the functional analysis and the pathway display. So this particular bioinformatic is what you need, or perhaps what you are going to discuss. So you need to understand what do you want, how are you going to discuss. All right. So to my conclusion, so I'm going to conclude. So amplicon and shotgun sequencing is basically an excellent approach to identify population within the ecosystem. Okay, so this particular metagenomic sequencing, it overcomes these laborious technique, especially when you want to sequence unculturable microorganism and you want to overcome cloning and sequencing, especially sequencing with Sanger. It takes a lot of time. So these are the plus point or the advantage of metagenomics. So to conclude, this is the feature of the sequencing which I discussed earlier. So application for amplicon and shotgun metagenomics. So for amplicon, it's very species specific, but for shotgun, it tend to focuses in all the species and functions in a sample. Amplicon sequencing, the technology will be Illumina, while shotgun will be Illumina and PacBio. You can choose either one. Okay, what can you discover with amplicon? You can discover species, but with shotgun, you can find novel species, you can find functionality and the relationships. Okay, so what kind of downstream analysis can you do? You can do a relationship study, like you can build phylogenetic trees and see what are the relationship between all these particular organisms. Shotgun, not only gene function analysis, you can also look into the pathways, okay, and also relationship that involves within the microbiome, which means you can look into bacteria, fungus, algae, and etc. So that's all for my presentation today for the webinar. So thank you so much for joining me today. So if you have any further enquiries, kindly email to ngs@apicalscientific.com. Thank you and see you in the coming webinars.